Hello, I am the Nerdy Hedgehog. Check out my channel every week to learn about the top research papers published in the biomedical sciences. You don't have to stop your lab work to stay up to date. I read the abstracts so that you don't have to. The first study was published in the journal Nature Medicine, entitled, Homozygous BCMA Gene Deletion in Response to Anti-BCMA CAR T Cells in a Patient with Multiple Myeloma. The abstract reads, B-cell maturation antigen, BCMA, is a target for various immunotherapies and a biomarker for tumor load in multiple myeloma (MM). We report a case of irreversible BCMA loss in a patient with MM who was enrolled in the KarMMa trial, NCT03361748, and progressed after anti-BCMA CAR T-cell therapy. We identified selection of a clone with homozygous deletion of TNFRSF17, BCMA, as the underlying mechanism of immune escape. Furthermore, we found heterozygous TNFRSF17 loss or monosomy 16 in 37 out of 168 patients with MM, including 28 out of 33 patients with hyperhaploid MM who had not been previously treated with BCMA targeting therapies, suggesting that heterozygous TNFRSF17 deletion at baseline could theoretically be a risk factor for BCMA loss after immunotherapy. The next study was published in the journal Nature Medicine, entitled, T-cell-engineered T-cells targeting E7 for patients with metastatic HPV-associated epithelial cancers. The abstract reads, genetically engineered T-cell therapy can induce remarkable tumor responses in hematologic malignancies. However, it is not known if this type of therapy can be applied effectively to epithelial cancers, which account for 80 to 90% of human malignancies. We have conducted a first-in-human, Phase 1 clinical trial of T-cells engineered with a T-cell receptor targeting HPV-16E7 for the treatment of metastatic human papillomavirus-associated epithelial cancers, NCT02858310. The primary endpoint was maximum tolerated dose. Cell dose was not limited by toxicity with a maximum dose of 1 × 1011 engineered T-cells administered. Tumor responses following treatment were evaluated using RECIST, Response Evaluation Criteria in Solid Tumors, guidelines. Robust tumor regression was observed with objective clinical responses in 6 of 12 patients, including 4 of 8 patients with anti-PD-1 refractory disease. Responses included extensive regression of bulky tumors and complete regression of most tumors in some patients. Genomic studies, which included intrapatient tumors with dichotomous treatment responses, revealed resistance mechanisms from defects in critical components of the antigen presentation and interferon response pathways. These findings demonstrate that engineered T-cells can mediate regression of common carcinomas, and they reveal immune editing as a constraint on the curative potential of cellular therapy and possibly other immunotherapies in advanced epithelial cancer. The next study was published in the journal Cell Stem Cell, entitled, Sequential CRISPR Gene Editing in Human IPSC Charts the Clonal Evolution of Myeloid Leukemia and Identifies Early Disease Targets. The abstract reads, human cancers arise through the sequential acquisition of somatic mutations that create successive clonal populations. Human cancer evolution models could help illuminate this process and inform therapeutic intervention at an early disease stage, but their creation has faced significant challenges. Here, we combined induced pluripotent stem cell, IPSC, and CRISPR-Cas9 technologies to develop a model of the clonal evolution of acute myeloid leukemia, AML. Through the stepwise introduction of 3 driver mutations, we generated IPSC clones that, upon hematopoietic differentiation, capture distinct premalignant stages, including clonal hematopoiesis, CH, and myelodysplastic syndrome, MDS, culminating in a transplantable leukemia, and recapitulate transcriptional and chromatin accessibility signatures of primary human MDS and AML. By mapping dynamic changes in transcriptomes and chromatin landscapes, we characterized transcriptional programs driving specific transitions between disease stages. We identify cell-autonomous dysregulation of inflammatory signaling as an early and persistent event in leukemogenesis and a promising early therapeutic target. The next study was published in the journal Science Translational Medicine, entitled, Engineering Adeno-Associated Viral Vectors to Evade Innate Immune and Inflammatory Responses. The abstract reads, nucleic acids are used in many therapeutic modalities, including gene therapy, but their ability to trigger host immune responses in vivo can lead to decreased safety and efficacy. In the case of adeno-associated viral (AAV) vectors, studies have shown that the genome of the vector activates toll-like receptor 9, TLR9, a pattern recognition receptor that senses foreign DNA. Here, we engineered our vectors to be intrinsically less immunogenic by incorporating short DNA oligonucleotides that antagonize TLR9 activation directly into the vector genome. The engineered vectors elicited markedly reduced innate immune and T-cell responses and enhanced gene expression in clinically relevant mouse and pig models across different tissues, including liver, muscle, and retina. Subretinal administration of higher dose AAV in pigs resulted in photoreceptor pathology with microglia and T-cell infiltration. These adverse findings were avoided in the contralateral eyes of the same animals that were injected with the engineered vectors. However, intravitreal injection of higher dose AAV in macaques, a more immunogenic route of administration, showed that the engineered vector delayed but did not prevent clinical uveitis, suggesting that other immune factors in addition to TLR9 may contribute to intraocular inflammation in this model. Our results demonstrate that linking specific immunomodulatory non-coding sequences to much longer therapeutic nucleic acids can cloak the vector from inducing unwanted immune responses in multiple, but not all, models. This coupled immunomodulation strategy may widen the therapeutic window for AAV therapies as well as other DNA-based gene transfer methods. The next study was published in the journal Science Advances, entitled, "Relaxin Gene Delivery Modulates Macrophages to Resolve Cancer Fibrosis and Synergizes with Immune Checkpoint Blockade Therapy." The abstract reads, "Cancer fibrosis serves as a major therapeutic barrier in desmoplastic tumors. Relaxin, RLN, a systemic hormone, is efficacious to decrease fibrosis, but the in vivo mechanism of action is not clear. Considering the localization of relaxin family peptide receptor type 1, RXFP1, the receptor for RLN, on macrophages, we hypothesize that macrophages can be modulated by RLN to ameliorate cancer fibrosis. Using KPC mouse model of pancreatic ductal adenocarcinoma, PDAC, here, we report locally expressed RLN with targeted gene delivery induces increased F4/80 plus C.D. 206 plus macrophages originating from Ly6C plus monocytes, promoting fibrosis depletion and cytotoxic T-cell infiltration. Moreover, RLN gene delivery synergizes with PD-L1 blockade for tumor inhibition by enhancing T-cell mediated tumor cell killing and macrophage phagocytosis. Collectively, our results reveal previously unidentified insights into the modulation of macrophages to regulate tumor-associated fibrosis, providing a feasible strategy to reverse the immunosuppressive environment and improve the therapeutic outcome of checkpoint immunotherapies. The next study was published in the journal Cell Stem Cell, entitled, "Dissecting the Impact of Regional Identity and the Oncogenic Role of Human-Specific Notch2NL in an HESC Model of H3.3G34R Mutant Glioma." The abstract reads, H3.3G34R mutant gliomas are lethal tumors of the cerebral hemispheres with unknown mechanisms of regional specificity and tumorigenicity. We developed a human embryonic stem cell, HESC, based model of H3.3G34R mutant glioma that recapitulates the key features of the tumors with cell type specificity to forebrain interneuronal progenitors but not hindbrain precursors. We show that H3.3G34R, ATRX, and TP53 mutations cooperatively impact alternative RNA splicing events, particularly suppression of intron retention. This leads to increased expression of components of the Notch pathway, notably Notch2NL, a human-specific gene family. We also uncover a parallel mechanism of enhanced Notch2NL expression via genomic amplification of its locus in some H3.3G34R mutant tumors. These findings demonstrate a novel mechanism whereby evolutionary pathways that lead to larger brain size in humans are co-opted to drive tumor growth. The next study was published in the journal Cell Research, entitled, "Formative Pluripotent Stem Cells Show Features of Epiblast Cells Poised for Gastrulation." The abstract reads, "The pluripotency of mammalian early and late epiblast could be recapitulated by naive embryonic stem cells, ESCs, and primed epiblast stem cells, EPISCs, respectively. However, these two states of pluripotency may not be sufficient to reflect the full complexity and developmental potency of the epiblast during mammalian early development. Here we report the establishment of self-renewing formative pluripotent stem cells, FPSCS, which manifest features of epiblast cells poised for gastrulation. FPSCS can be established from different mouse ESCs, pre/early gastrular epiblasts and induced PSCs. Similar to pre/early gastrular epiblasts, FPSCS show the transcriptomic features of formative pluripotency, which are distinct from naive ESCs and primed episcs. FPSCS show the unique epigenetic states of E6.5 epiblast, including the superbivalency of a large set of developmental genes. Just like epiblast cells immediately before gastrulation, FPSCS can efficiently differentiate into 3 germ layers and primordial germ cells, PGCs, in vitro. Thus, FPSCS highlight the feasibility of using PSCs to explore the development of mammalian epiblast. The next study was published in the journal Small, entitled, Immune-Stimulating Antibody Photosensitizer Conjugates via Fc-Mediated Dendritic Cell Phagocytosis and Phototriggered Immunogenic Cell Death for KRAS-Mutated Pancreatic Cancer Treatment. The abstract reads, although cetuximab, CTX, is a chimeric epidermal growth factor receptor, EGFR, antibody, the anti-tumor efficacy of CTX has a negligible effect in patients with Kirsten-Rat sarcoma 2 viral oncogene homolog, KRAS, mutated pancreatic adenocarcinoma. Given that all extant treatments are ineffective due to the undruggable characteristics of KRAS-mutated cancer, alternative strategies have been investigated. In this work, CTX conjugated maleimide polyethylene glycol E6, CMPC, is designed to strengthen its anti-tumor efficacy. With strong affinity for EGFR overexpressing ASP1 cells, CMPC with laser exerts the greatest cytotoxicity, 90%, and induction of immunogenic cell death. Through a combination of fragment crystallizable region-mediated antigen uptake by CTX and danger-associated molecular patterns released by photodynamic therapy, PDT, phagocytosis and maturation of dendritic cells treated with CMPC plus laser show dramatic increases. In vivo biodistribution and anti-tumor effect also demonstrate that CMPC has significant tumor selectivity and tumor ablation efficacy upon laser irradiation. Furthermore, a large number of CD4+, CD8+ T cells and mature DCs and natural killer cells are infiltrated in CMPC with laser-treated tumor tissues and tumor-draining lymph nodes, revealing both innate and adaptive cellular immune stimulation. This synergistic effect with CMPC and laser treatment provides an effective approach for pancreatic cancer immunotherapy attributed to both CTX and PDT. The next study was published in the EMBO Journal, entitled, Nuclear IL-33/SMAD Signaling Axis Promotes Cancer Development in Chronic Inflammation. The abstract reads, interleukin, IL-33 cytokine plays a critical role in allergic diseases and cancer. IL-33 also has a nuclear localization signal. However, the nuclear function of IL-33 and its impact on cancer is unknown. Here, we demonstrate that nuclear IL-33-mediated activation of SMAD signaling pathway in epithelial cells is essential for cancer development in chronic inflammation. Using RNA and ChIP sequencing, we found that nuclear IL-33 repressed the expression of an inhibitory SMAD, SMAD6, by interacting with its transcription factor, RUNX2. IL-33 was highly expressed in the skin and pancreatic epithelial cells in chronic inflammation, leading to a markedly repressed SMAD6 expression as well as dramatically upregulated pSMAD2/3 and pSMAD1/5 in the epithelial cells. Blocking TGF-beta/SMAD signaling attenuated the IL-33-induced cell proliferation in vitro and inhibited IL-33-dependent epidermal hyperplasia and skin cancer development in vivo. IL-33 and SMAD signaling were upregulated in human skin cancer, pancreatitis, and pancreatitis-associated pancreatic cancer. Collectively, our findings reveal that nuclear IL-33/SMAD signaling is a cell-autonomous tumor-promoting axis in chronic inflammation, which can be targeted by small molecule inhibitors for cancer treatment and prevention. The final study was published in the journal Advanced Materials, entitled, Functional Skeletal Muscle Regeneration with Thermally Drawn Porous Fibers and Reprogrammed Muscle Progenitors for Volumetric Muscle Injury. The abstract reads, skeletal muscle has an inherent capacity for spontaneous regeneration. However, Recovery after severe injuries such as volumetric muscle loss, VML, is limited. There is therefore a need to develop interventions to induce functional skeletal muscle restoration. One suggested approach includes tissue-engineered muscle constructs. Tissue engineering treatments have so far been impeded by the lack of reliable cell sources and the challenges in engineering of suitable tissue scaffolds. To address these challenges, muscle extracellular matrix, MEM, and induced skeletal myogenic progenitor cells, IMPCs, are integrated within thermally drawn fiber-based microchannel scaffolds. The microchannel fibers decorated with MEM enhance differentiation and maturation of IMPCs. Furthermore, engraftment of these bioengineered hybrid muscle constructs induced de novo muscle regeneration accompanied with microvessel and neuromuscular junction formation in a VML mouse model, ultimately leading to functional recovery of muscle activity. Thank you for listening. Please like and subscribe to stay informed on the latest research.